Hello, my name is Carol Leibel, and today we're going to examine the second part to protein synthesis, translation. We are finally going to make that protein. This is a little video that is an overview of protein synthesis. Now, I've got to admit that this is really aimed at first-year biology students, but it gives you a nice overview and a nice reminder of what happened in the process of transcription. So let's watch this video. Oh, and by the way, enjoy the music.
Provides a series of signals, or a code, that directs the production of protein. The letters of the code correspond to the bases along the double helix. C for cytosine, G for guanine, A for adenine, and T for thymine. Each protein molecule has its own code, determined by the sequence of these bases. Beginning at an active site, a group of enzymes binds to the DNA molecule. This enzyme group is able to read the coded message. Part of this group travels down one strand of the helix. It chemically recognizes the bases as it passes and matches to them other nucleotides from the surroundings, creating a new molecule. This is done according to the correct pairing rules. C with G and A with U, where U stands for uracil, which replaces thymine in this new molecule. The new molecule is ribonucleic acid, or RNA. RNA is an intermediate step in the production of protein. These molecules of RNA are called messenger RNA because they carry the transcribed message to another part of the cell outside the nucleus to be decoded. The messenger RNA travels to a ribosome. It is here that proteins are actually manufactured. The ribosome reads off the message from the RNA in three-letter groups called codons. The codons are the words of the message. Each codon is matched to a specific amino acid, which is brought in and attached in sequence. As the codons are read off across the ribosome, the protein molecule that is being formed grows longer. The last three-letter group codes for the termination of the process, and the sentence, in the form of a protein molecule, is completed.
So, how did you enjoy that video? I thought the music was just outstanding, but nonetheless it does give you some idea about the process of protein synthesis that involves two parts: transcription, using a DNA template to make a piece of RNA, and then taking that RNA molecule and using that as a template to direct protein synthesis. Alright, so let's go on about translation. Translation involves messenger RNA being used as a template to make a protein molecule. Remember, the word template means a pattern. The materials that you're going to need for translation includes a ribosome, transfer RNA with the correct amino acid connected to it, and mature messenger RNA that has been processed in the nucleus if you're referring to a eukaryotic cell. So let's talk a moment about the ribosome structure. First off, ribosomes are made from proteins and ribosomal RNA. They have two subunits, a large subunit and a small subunit. There are three sites where the transfer RNA can attach. We have the A site, and that's where the transfer RNA is going to arrive with its amino acid. We have the P site, where the transfer RNA sits next to a second transfer RNA, where a covalent bond is formed between the amino acids, and that is right there. And then you have the all-important E site. And this was only discovered just recently. And that's the site where the transfer RNA is going to leave. However, when it leaves, it exits and it leaves behind the amino acid. Now, one thing I would like to point out is that prokaryotic and eukaryotic ribosomes, while they are similar because they both have a small subunit and a large subunits with the E, P, and A sites, they are slightly different from one another. And that difference has allowed antibiotics to interfere with the functioning of prokaryotic ribosomes while leaving eukaryotic ribosomes intact. The second thing that we need in the process of translation is a transfer RNA. Now, what we're looking at here on this slide is, first off, a two-dimensional structure of a transfer RNA. You'll notice that over here is the 3' end and here's the 5' end. And if you were to follow it along, you would see that it is indeed a single molecule. However, it does allow for base pairing among its nucleotides, and it forms this cloverleaf structure, if you will. Now, down here at the bottom is a very important part of the transfer RNA. It's called the anticodon. And this is the part that is complementary to the codon. So, if on the messenger RNA it reads UUC, the anticodon is going to be AAG. And then up here at the top is a place where it will attach an amino acid and bring the correct amino acid according to the codon. So this right here is called, because it's complementary and seems to be the opposite, if you will, it's called an anticodon to the codon. Now, this two-dimensional cloverleaf structure doesn't stay flat. Instead, what happens is that it folds upon itself, forming a three-dimensional structure that looks almost like an L. So right down here is the anticodon, and then over here is where we would find the attachment of the amino acid. Now, theoretically, there should be 61 transfer RNAs, because remember, there are 64 codons, but 3 of them are stop codons. But it turns out that there's often redundancy at position 3 in the messenger RNA codon. And so the base pairing rule at position number 3, right here, is not as strict. So, for example, U on the anticodon here, if this were a U over here, it can bind to U, which is normal. However, the other odd thing is that it could also bind to G. And some transfer RNAs include a modified form of adenine, and that modified form of adenine is called inosine, and it turns out that inosine can bind with U, C, or A on the codon. Well, again, theoretically, there should be 61 of these transfer RNAs, but because position number 3 is a little more relaxed, it turns out that there are only 45 transfer RNAs used. And that's, by the way, that's called the wobble effect. Alright, this next slide gives you a three-dimensional viewing of a transfer RNA. Kind of cool. Alright, so here's an example of the wobble effect. UCA and UCG, okay, code for serine, okay? UCA and UCG code for serine. However, the U will base pair with either an A or a G, so the transfer RNA will still bring the amino acid serine. So in this case here, there's only one transfer RNA for both of those codons, and it still will bring serine to the correct place. Another example, GCA and GCG code for alanine. Okay? However, the transfer RNA, the anticodon, the U will base pair with either an A or a G, so the transferrin A will still bring the amino acid alanine. Kind of cool. So in reality, what that means, again, instead of 61 transferrin A's needed, we find that there's only 45. Alright, so now, is that we have to be able to put the correct amino acid onto the transferrin A. And the transferrin A gets its amino acid by combining with a charging enzyme, which is called aminosyl transfer RNA synthetase. This amino acid will put the correct, this enzyme will put the correct amino acid onto the transfer RNA according to its anticodon. And in addition, in order to make this work, you have to have ATP. So here is aminosyl tRNA synthetase. Alright, here you can see right here, there's the active site, and it is specific for a particular amino acid. So here comes the amino acid. You have to have a phosphate group, so ATP is needed. And so now, once that amino acid is in place, the transferrin A comes in. And now the enzyme will combine the amino acid to the correct transfer RNA. And now that transferrin A is said to be activated. So it turns out that the parts of translation are somewhat like the parts of transcription. It involves initiation, or the beginning, elongation, and termination. So in initiation, what's going to happen is there's going to be a small subunit attaching to the messenger RNA at the codon AUG. Okay, now what's going to happen next is that a transfer RNA with the anticodon UAC will attach at that P site and will bring with it the amino acid methionine. Alright, so theoretically, all proteins should start with methionine, but once the protein is made, modifications take place, and quite often this is going to be removed. Alright, so there is that transferrin A. Now what's going to happen next is that the large subunit is going to attach, and that's going to require some phosphorylation involving guanine triphosphate. And so now the large subunit attach, and the transferrin A is sitting here at the P site. And now initiation is complete. Alright, part 2, elongation. And it's almost cyclic in nature. So the next thing that's going to happen is there's going to be a transferrin A that's going to arrive at the A site. And it's going to be complementary to that messenger RNA, and it's going to have the correct amino acid. So now the two are sitting side by side. What's going to happen is a dehydration reaction in which water is going to be removed, and we have a covalent bond or a peptide bond being formed. Once that occurs, the transferrin A is going to release that amino acid. And so now, here's the polypeptide chain right there on that amino acid. And translocation is going to take place. And again, a phosphate is needed, so guanine triphosphate is involved. Translocation is going to take place. And these two are going to move down to the next site. So this transferrin A is moving to the E site, and this transferrin A is moving to the P site, and now this transferrin A is going to exit the ribosome. And now it's going to look for another aminosyl transferrin A synthetase. And now we're ready for the cycle to start all over again. And now the A site is ready to receive another transferrin A. However, that transferrin A must be complementary to that codon. Alright, so this process continues and the polypeptide chain grows. And finally, we come to the termination. Remember we said that there were 3 stop codons. Okay, and they could be UAG, UAA, or UGA. And what happens is that there is a protein that recognizes these stop codons, and it's called a release factor. And so what will happen is that protein will come in and it will attach the stop codon. When that happens, it causes the release of that last transfer RNA, and when that happens, it also disengages from that polypeptide that's being made, and so now the ribosome will come apart and release that messenger RNA. That messenger RNA can be translated again and another protein can be made. Alright, so now we want to examine what happens to proteins that are destined to be exported from the cell. Well, first off, those proteins, as they're being made, the very beginning of that protein has what we call a signal peptide. And that little signal attracts something called a signal recognition particle, which is made out of protein and RNA. It attaches to that little signal, and so now this little complex is attracted to the pore in the endoplasmic reticulum, and it kind of pulls this whole complex over. And as it does that, what's going to happen is that the polypeptide chain is going to be threaded into the cisternae, or the inside of the endoplasmic reticulum. Now once the protein is made and threaded inside, that signal part of the protein is removed, and now what's going to happen is that the messenger RNA and the ribosomes are going to disengage, and now the protein is going to be on the inside of the endoplasmic reticulum. As you know, what's going to happen next is that this is going to be packaged up into a little vesicle, and now it's going to go to the Golgi apparatus to be processed and finally be packaged and tagged for export outside of the cell. We want to take some time and examine what happens when there are mutations in the gene and the effects on its protein. So there are three types of what we call substitution mutations. So let's suppose that right here we have a gene, and so let's suppose that right here we have a mutation, and instead of GGU we put an A right here. Well, if it is now instead of a C, it's GGU, what happens is that GGC and GGU, they both code for glycine. So even though there is a mutation in the DNA and there's a U there instead of the C, there's no effect on the protein. So the protein has the same structure, nothing happens. Alright, so now let's suppose that now at this position, instead of a G, we have an A. So instead of being GGC, it is now AGC, and that does make a difference. This now is going to code for serine. The question is, does having serine there instead of glycine make a difference? It depends on where in the protein this mutation takes place. Sometimes it has an effect on the protein structure, sometimes it does not. If it does not, then that protein is just said to be an isoenzyme or an isoform of that same protein. And they are indeed two different proteins, but the function of the protein does not change. Sometimes, though, having that one little change does indeed make a difference. For example, cystic fibrosis and sickle cell anemia both occur because of one little change right there. Well, at least one substitution, if you will. And so it does indeed make a different protein with different properties, and it doesn't function the same way. Alright, so and by the way, that's called a missense mutation. Now, a nonsense mutation is one in which you have a change, like right here, where we have lysine, it is AAG. If you have a mutation and now you put a U there instead of that A, that will cause a stop codon. So now your protein is methionine, stop. That makes no sense at all, if you will. And so that's called a nonsense mutation, and it causes the shortening of the polypeptide chain. Most of the time that's a very, very detrimental mutation. Okay, the next type of mutation that's important is called frame shift mutations. And frame shift mutations result from either an insertion or a deletion of a nucleotide. So, for example, this first one, we have a messenger RNA, and it's asking for methionine, lysine, phenylalanine, and glycine. And what we're going to do is right here is we're going to remove that U. Now, what happens when we remove that U is that now the rest of the protein is going to be off, if you will. The rest of the protein is going to be completely changed. This is called a missense mutation. A change in one amino acid is going to change the sequence for the rest of the polypeptide chain. Okay? Alright, now remember we have mentioned that the typical polypeptide chain is 400 amino acids long, and that we mentioned that genes typically may be 8,000 nucleotides long or so forth. And so if you keep deleting or inserting until you get 3, what will happen is that's going to put it back on track, if you will. Alright? If you have 3 insertions or 3 deletions at the same time, okay, like this one right here, then you're only going to miss 1 amino acid, and that's not going to perhaps be so detrimental. Alright, you can indeed also have a nonsense mutation if you turn around and you insert something that turns around that will cause you to have a stop codon. So it is indeed possible in a frame shift mutation to have missense mutations and nonsense mutations. And again, how they affect the protein depends upon which amino acid is being deleted or changed around or what have you. An example of a frame shift mutation that is detrimental is Tay-Sachs disease. This is an autosomal recessive disorder marked by a degeneration of both mental and physical capabilities. At its worst, death usually occurs around the age of 4, although both juvenile and late onset forms of the disease have been observed. It's caused by a buildup of a particular lipid in the brain that's normally broken down by an enzyme called hexoaminidase. Although different mutations in the same gene can lead to the disease, in most cases a 4 base pair insertion in the hex gene renders a premature stop codon, leading a profound deficiency in one of the subunits of the hexoaminidase protein. So I do want to remind you that not all frame shift and point mutations are detrimental. Some of them can lead to new genes and novel proteins that would be beneficial to the organism. After all, it's the ability to change DNA that leads to genetic adaptations and ultimately evolution of a species. It's important to relate this back to evolution, and that this is the raw material upon which evolution works. Alright, so this is a nice review of the process of translation.
In translation, a cell reads the message in a messenger RNA molecule, mRNA, and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when a small ribosomal subunit binds to an mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine, abbreviated MET. A large ribosomal subunit binds to the small one, creating a functional ribosome. The initiator tRNA sits in the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the P site exits from the ribosome. The ribosome then translocates, or moves, the tRNA in the A site, carrying the growing polypeptide to the P site. Meanwhile, a new tRNA brings its amino acid to the A site, and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. The completed polypeptide detaches from the tRNA that is in the P site, and the components come apart. The polypeptide folds and may be modified or combined with other polypeptides to form a functional protein.
Alright, so a couple of things I need to point out is that you'll notice that on the ribosome they did not have the newly discovered E site, and when it came to termination they did not show you the protein termination factor that combines with those three different codons. So this is the end of translation. You've got a functional protein now, and life is good. So take care, and I hope to see you later. Bye!
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Alright, so I hope you enjoyed that review of protein synthesis. And so now I'm really ready to say goodbye. Have a good day, and I hope to see you again or hear from you again. Take care. Bye.